So today we're going to pick up where we left off, and we're going to start amino acids, peptides, proteins, and isoelectric point, section 3. So a basic overview of today's recorded lecture. We're going to pick up with calculating isoelectric point, discuss some basics about basic peptides, some comments about shape and composition of proteins and how amino acids come together. And then we're going to go to secondary structures and what I'm referring to as beyond secondary structures or super secondary structures. So for this portion of the lecture, if you're pretty comfortable in calculating isoelectric point, you might as well skip forward, or take the time here to maybe pause on a slide and practice calculating the isoelectric point of each of these slides here. So, to start off with something simple, we have here, consider the following dipeptide, right? So we have our first amino acid, right, carboxyl group, and then the amino group of the next amino acid. So, one amino acid, two amino acids, right? So dipeptide. You want to begin by calculating what its isoelectric point actually is. So to do this, I suggest start by drawing the amino acid or the peptide, dipeptide in this case, right? Here it's already drawn for us, but if for practice and for practice's sake, I recommend drawing it. That way you can identify the amino acids, their specific pKa's, and then you can also identify the specific pKa's of each amino acid. So what's going to pop up next? So if we look at these two amino acids, the first guy here is glycine. So the only ionizable protein on glycine here is our our amino group, and then our other amino acid here, methionine, has again only one proteinizable functional group. In this case, the COH. So we have a COH, or the C terminus end of methionine, and then over here NH3 plus, or the N terminal end of glycine, right? And so if we refer to our chart, we know the pKa of that amino group is 9.6, and the pKa of that methionine group, or excuse me, the pKa of that carboxyl group is 2.28, right? So we have identified the amino acids here, and we've identified the pKa's of the ionizable functional groups. So the next step is to go through the protonation state of each of these functional groups, pH by pH, right? So in class, I recommend doing this by essentially normally starting at 1 is pretty safe. So you could start at a pH of 1, draw your dipeptide in this case, and remember that when you are to the left of a functional group or an ionizable ionizable proton with a specific pKa, if you were to the left of it on its titration curve, it would be protonated. So, looks like yeah, okay, so let's start with 1. Okay, so at a pH of 1, we know that a pH of 1 is well below pKa of 2.28, so this guy would be protonated and have 0 charge, and we are very, very far to the left of the pKa of this guy. So, they are both protonated. This is 0 charge, this is a positive 1 charge, so at a pH of 1, the overall charge on our dipeptide here is +1. And then next, I recommend picking a pH in between the two pKa's here, right? There's only 2, so you only have to pick 1 pH. If there was 3 pKa's, you would have to pick a pH in between each set of pKa's. I believe we'll see that on the next example. So, let's pick a pH of 5. At a pH of 5, now we are to the right of our carboxyl group here, so he will be unprotonated, as he is in this image, and have a -1 charge, and we are still to the left of our amino group over here, so he's also still protonated. So that gives us a -1 and a +1, which means at a pH of 5, our dipeptide here has a net charge of 0. So for our last pH, let's just for for the sake of the practice and the sake of this question, use a pH of 12. So at a pH 12, we are still far, far to the right of the pKa of our carboxyl group. So we know this guy is still unprotonated. And now we are to the right of our amino pKa here as well. So he will donate his proton to the solution around him and no longer be protonated. So now, after doing that, he has a 0 charge. The carboxyl group has a -1 charge, so the net charge on our dipeptide is -1, which means at a pH of 5, we have our Zwitter ion, and there is a net 0 charge on our dipeptide, right? So after doing that, all we have to do is take the average of our pKa's that straddle that pH, and do I have that? It comes up. I believe it comes up on the next slide. So you really just take the average of this. So you add 11.88 divided by 2, and you should get roughly 5.5. So following up from the previous slide, right, here's our pKa's. We know a pH of 5 is in between here. So add and just divide, and turns out our isoelectric point is 5.94, right? Okay, and then the last part of this question, in which direction, and so you'll have to do this on your own, in which direction will the dipeptide move at a pH of 1, 3, 5, 7, 9, and 12? So go through the same process we did in the previous slide. Draw our dipeptide here and label its overall charge at a pH of 1, 3, 5, 7, 9, and 12. And then recall which direction ions move in an electrolytic field, right? So remember, in an electrolytic field, anions, or molecules that have a negative charge, move towards the anode. So the anode is positive in this situation, right? So anions, negative ions, travel towards the positive pole or the anode. And then vice versa, cations travel towards the cathode. So ions that have a positive charge will travel towards the negatively charged pole. Right? So please review this. This is about as simple as these examples can really get. There's only 3 pHs to attempt to try. And really practice drawing your molecules and then figuring out whether your functional groups donate or receive hydrogens from the surrounding environment based on the pH and pKa of those functional groups. So now let's look at a tripeptide, right? So in this example, we have 3 amino acids. So if you're used to looking at these guys now, N-terminus of our first amino acid, C-terminus of our first amino acid, there's that peptide bond. Here's the second peptide bond. And we have 1 amino acid, 2 amino acids, and our third amino acids. If you're not used to figuring out if you're not used to being familiar with amino acids based on their structure, maybe take a moment to pause the video here and try and figure out what these guys are before the slideshow advances. Okay, I am now going to advance. So our first amino acid here is valine, and in valine we only have 1 protonizable group, so that's our NH3. So the pKa of this is 9.62, right? Amino group, 9.62. Our next amino acid has no protonizable functional groups, and this is actually methionine, same as the previous slide. But again, no protonizable functional groups here, so nothing to worry about there. And then our last amino acid over here is tryptophan. And in tryptophan, we only have, again, that carboxyl group with a pKa of 2.83. So again, even though we have 3 amino acids now instead of 2, we only have 2 ionizable functional groups, which are the targets you're looking for when trying to figure out how to calculate the isoelectric point of a single amino acid or a dipeptide, or again, in this case, a tripeptide. So, as in the previous slide, I recommend picking, a pH that is far more acidic than both functional groups, far more basic than both functional groups. So again, a pH of 1 will work here. That's below both of these pKa's, and then a pH of, say, 14 will also work because it's well above both of these pKa's. And then you pick a pH in between these two pKa's. So maybe, again, take a moment to pause the video here, draw out our molecule, and then try and figure out whether at a pH 1 this guy is protonated and this guy's the protonation state of both these ionizable hydrogens, right? I'm not going to try and give it away if you're about to pause the video. So go ahead and pause, test at a pH of 1, is he protonated, is he protonated, pH of 5, is he protonated, is he protonated, and then at a pH of 14, is he protonated, is he protonated. Go ahead and do that now. So, if you took the time to do that exercise, we know that again, at a pH in between our pKa's, again, this is a pretty simple example, even though it has 3 amino acids, at a pH in between both of these pKa's is where our isoelectric point is. So, again, we just add those up, take the average, and the isoelectric point of this guy, this tripeptide, is 6.225. If you want more practice, please also see the posted video about how to calculate an isoelectric point. It walks through the whole process step by step with slightly more complicated examples. But it also demonstrates how to draw the table I use to solve these equations and these questions. So if you're feeling a little bit lost and still not, this still isn't quite making sense, go ahead and pause here and check out that other video. Okay, so for our last example, this is more complicated than the previous two examples, but still not too complicated if you just break down the problem as I have demonstrated before, right? So break it down into these steps. In order to calculate the isoelectric point, first start by drawing your amino acid or your peptide, right? So go ahead and draw this on your own sheet of paper, or maybe just make note of what the ionizable groups are on these amino acids. So this short peptide contains alanine, glutamine, glycine, and lysine, right? So when strung together, alanine has a protonizable amino group. Glutamine, or glutamic acid over here, has a ionizable R group with a carboxylic acid, so you'll have to look on our chart to find the pKa for this, or I'll actually give it to you in the next slide. Glycine, with his single hydrogen R group, has no ionizable groups. And then lysine over here has both an R group with an ionizable amine or amino group and a C-terminus carboxyl group here that can donate and accept hydrogens from the surrounding solution. So, again, start by drawing your amino acids or your peptides. Identify your amino acids and the pKa's of each of these. So if you haven't done that, feel free to pull up your table, check that out, and then once that's all said and done, one last part of the exercise is to test or predict which way our peptide is going to move in an electrical field at each of our pHs. So I'm going to advance the slide now. If you'd like to, take the time to maybe look up these pKa's on your own. Otherwise, feel free to follow me right along. Okay, so here it is. Similarly to our example video, I have drawn our table that takes into account the ionizable functional group of each of our amino acids here. I have our designated pHs to test our charges at, right? So 1 is below the pKa of all functional groups, 13 is above the pKa of all functional groups, and then I have pHs to test the protonation status of each pKa in between each pKa. So again, I recommend doing this for every problem you have. It's the safest way to go about getting the correct answer every time. So if you look through the chart real quick, you'll notice that at a pH of 1, we have positive 2 from the protons still on those amino groups. And then as we move up, our carboxylic acids donate their hydrogens and become negative. And, as we move further and further up the pH scale here, eventually both of our carboxylic acids have donated their hydrogens, while our amino groups have also now donated their hydrogens. But the carboxylic acids, once they donate their hydrogen, carry a negative charge, and the amino groups, once they donate their hydrogen, carry 0 charge. So if we compare pHs, we notice that at a pH of 7, We have -1 here, -1 here for lysine and glutamic acids functional groups, and then +1 on alanine and +1 on glycine amino groups, which leaves us at a net charge of 0. So if we look at the pKa's that straddle that net charge of 0, flying through these, we notice that the pKa above the 0 is 9.7, so the amino group from alanine, and the pKa below that 0 is 4.3 from the carboxylic acid group here on glutamic acid. Right, so all we have to do is add those up, take the average, and we arrive at an isoelectric point of exactly 7. Go ahead and continue to practice these if they are still confusing. So the purpose of this slide is just to discuss and demonstrate that not all peptides have to be incredibly long or complicated to have important roles, right? So I know in class I've mentioned that some peptides or proteins can be 500 amino acids long or even larger. Again, this slide is really just meant to demonstrate that you don't need a big complicated chain of amino acids to make a peptide with an important function. So the first example here, glutathione, is only 3 amino acids long, right? If we look over here, here's one of our amino acids, a second, and a third. And if you note, there's something odd about the way this peptide is strung together. But we'll talk about that later throughout the semester. And glutathione has a big job, right? It's involved in some detoxification processes, involved in oxidative stress responses, and a number of things. And again, it's only 3 amino acids long, and it has an incredibly important function. So the second one on our list here, atrial natriuretic factor, or ANF, helps block water reabsorption, right? And he's down here. So we can see that obviously this guy is longer than our first example, glutathione, but relatively short in the grand scheme of things. And we'll note that on this peptide, as well as 2 of our other examples, we have the formation of disulfide bonds between cysteine amino acids, emphasizing the role that covalent interactions between amino acids has on the overall structure, which leads to the overall function of many peptides. So our next example, vasopressin, actually stimulates water reabsorption in kidneys in response to low blood pressure. And the guy's right here, he's only about 9 amino acids long. And then oxytocin, which some of you may have heard of referred to as the love hormone or the love peptide protein, is right up here and also only has roughly 9 amino acids. And just a small note that oxytocin has much broader role than promoting relationships and things like that. I'm happy to discuss this more in class if you want to, but And then my last comment, when I get the chance, I like to reflect on some of the pharmacology I've been involved in the past. Endogenous opioids are also fairly short, right? Endogenous opioids are responsible for our body's endogenous pain management system, as well as some of the reward circuitry that goes on in our brain when we eat things like sugary food or fatty food, or win a competitive basketball game or football game. And these guys are also fairly short in the grand scheme of things, but play a huge role in a lot of the things that go on in learning, behavior, and again, endogenous pain management. So this slide is just a stop check to review. So just to review, go ahead and pause if you want to. So just a reminder, amino acids are bonded together with which bond? That would be the peptide bond between the carboxyl group of one amino acid and the amine group of the next. A polypeptide is a string or polymer of multiple peptides, right? So polypeptide would imply multiple peptides. And this string is the primary sequence built based on what? This string of amino acids is built on the basis of our DNA coding, and DNA is translated into, transcribed into RNA, and our RNA is translated into amino acids by their codon sequences, right? If this is sounding a little unfamiliar, be sure to review our video about the central dogma of biology. And then one last note about the unique unique restrictions that the peptide bond places on a string of amino acids, or a string of amino acids that leads to peptide or polypeptide structure. The rotation in a polypeptide is limited to the alpha carbon, right? So that peptide bond actually that forms has a little bit of resonance structure, which gives it a quasi double bond, which makes it stronger, but it restricts the rotation around that carbon and nitrogen molecules. So that adds strength to the peptide bond, but it limits the rotation that polypeptides or amino acids have around that alpha carbon. And if this isn't ringing a bell, check out our slide deck from when we first introduced and discussed the formation of the peptide bond. So this slide is meant to give an overview and a characterization of some of the classes of enzymes that we'll be talking about in class, right? And the overall functions of these classes of enzymes. Proteins, excuse me. Enzymes is one of these classes. So catalysis, right? Proteins, one of the families and functions of proteins is to perform catalysis. Usually this is performed by enzymes or organic catalysts. Some proteins add structural support, so things like collagen and elastin. Some proteins are actually involved in, making us move, right? So movement on the large scale with like muscle movement. You can review chapter 22 of our Maisfield and McCoy textbook to see some information about that. And then, on a much smaller level, I like to refer to our tiny little cytoskeleton railroads, dynein and kinesin, right? These guys are called motor proteins, and they literally walk down the cytoskeleton found within cells to help transport things. So some proteins are involved in defense, right? Adaptive and non-adaptive immune response. Others are involved in blood clotting. Some proteins are involved in regulation or long distance, regulation, and then one example of that is like long distance communication by hormone signaling. Cellular transport, so cellular or system transport in this case, right? So across cellular membranes, just moving ions across the cellular membrane sometimes requires a specialized protein, or moving oxygen through our blood systems, right? Hemoglobin is responsible for doing that, and so in this case it helps with gas exchange and gas transport. Some proteins are involved in storage, so macromolecular depots for development. Other proteins still are involved in the stress response, right? So xenobiotic degradation, so both good and bad. This is breaking down of foreign substances in your body, so like drugs or bacterial infections and stuff. Heat shock proteins, or HSPs, can actually activate and help chaperone and move proteins around your cell in response to things like heat shock. And you can consider those responses to stress responses. Other animals or other kinds of proteins, produced by other animals, would be a class of proteins called toxins, right? So I always put this little reminder in every semester: if you bite it and you die, it's poisonous, or you eat it. And if it bites you and you die, it's venomous. So things like frogs and mushrooms are technically poisonous, whereas things that sting you, like jellyfish and pufferfish, are venomous. But some toxins are made out of proteins, and they can be pretty nefarious. And then, as I've mentioned in class before, there's not a lot of black and white in biochemistry. So some proteins or enzymes even have multiple functions, right? You wouldn't be able to characterize them into just one little group. So one enzyme, glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, has multiple functions that you can see down here, right? So it oxidizes glyceraldehyde 3-phosphate, and we'll talk about this in glycolysis, but it can also be involved in DNA synthesis and repair, microtubule bundling, apoptosis, tRNA transport, and the list continues. And then if you look over here to the right, we've discussed motor proteins. I've showed this GIF before. We've talked about GFP, right? Some proteins even have evolved over time to arrange themselves in a way that they fluoresce, or illuminate, right, or luminesce, right, luciferase from fireflies, and then these examples here. So a note here about some nomenclature. So just to differentiate between configuration and conformation, right? So a configuration refers to the covalent structure. So to change the configuration of a molecule, a covalent bond must be broken and or formed. So we can think about this as the linear sequence of amino acids that are all covalently bonded together, right? Whereas a conformation refers to the non-covalent arrangement of atoms and molecules relative to one another. So this would be, when molecules adopt one or more conformations when dissolved in a solvent, or perhaps they're put into a hypotonic or hypertonic solution, or we've changed the pH of a protein, right? So conformation in this essence with respect to proteins would refer to how the three-dimensional shape of a protein changes in different kinds of solutions, or if a post-translational modification has occurred. Things like that change the conformation of a peptide or polypeptide. And then over here, so up here would be the linear sequence of amino acids, right? The configuration of all of our amino acids, and then the conformational change that occurs between linear sequence and of our folded protein would be down here, change in conformation. So following along with some of our nomenclature, right? One way to refer to proteins is, or are, as simple proteins, right? So this is only the string of amino acids. So some functional proteins that only have a simple structure, right? So things like serum albumins or keratin. Whereas another way to describe proteins is a conjugated protein, right? We've talked about things that can be conjugated to proteins. Maybe take a pause here and reflect on some of the different types of conjugations we can do to proteins, right? Do you remember any of the different things we can do to a protein? And as I usually do, I add in this example here, right? And this is conjugating a protein typically occurs after translation, right? Or otherwise known as post-translation. So maybe take a second to pause here again and review the types of post-translational modifications. So, again, we've discussed these, so I won't spend too much time talking about them. Right? A string of amino acids can be combined with a prosthetic group or a small covalent structured molecule that changes its shape or gives it extra function. Hemoglobin is an example of this, right? The heme group or heme prosthetic group is added to that string of amino acids to allow it to carry oxygen. Some additional nomenclature, right? So if a protein requires conjugation to have full function, before that conjugation occurs, prior to combination, these proteins are known as apoproteins and are incomplete, right? They won't have their full function. Whereas following combination with those post-translational modifications, they're known as a hollow protein, or closer to completion, or a whole protein, right? One way to remember what a hollow protein is just to simply think of it as a whole protein, right? It's been modified and now is whole and functional. So just to quickly review some examples of those, right? So remember, glycoproteins have carbohydrates added to them. Hemoproteins have heme groups. Lipoproteins have lipid molecules added to them. And then still other proteins have inorganic cofactors added to them, right? Like metalloproteins and phosphoproteins. So now here we will list the hierarchy of protein organization, right? So we have primary structure, which is simply the linear amino acid sequence of a protein, right? So that's literally which amino acids are strung together and bonded together. And if you pull them out into a string, what's the linear code that's been built. Following that, we have secondary structures. So these are considered, to be the backbone of larger structures of polypeptides. and are a result of conformational changes of that linear sequence, right? So more than likely you've heard of these before. An example would be the alpha helix or beta sheets. We'll talk a little bit more of those on the next slides. And then following that, we have tertiary structure, right? So this is when potentially a protein has completely folded into a functional shape. Multiple secondary structures have come together and created a three-dimensional protein with function, right? And this sometimes, again, this requires the formation of disulfide bonds between amino acids like cysteines, or through the addition of prosthetic groups or other post-translational modifications, right? Sometimes we need to add a little bit of something to that primary sequence to aid in the folding process of proteins to give them function. And lastly, quaternary structure, right? So this is when multiple polypeptides come together and form large assembled subunits with function, right? We'll talk about some of these in class, some large complexes of polypeptides that need to come together to have full function. So, some examples of secondary structures. Again, probably two examples that you guys have heard of before. The alpha helix, right? So one chain of amino acids, which can form into a helical shape. And we'll talk a little bit about how these form and how they're stabilized. The beta sheet, which is usually pretty flat. So here's an example, right? The backbone of our amino acid can come up and loop around and stick together here. Again, we'll talk about this in a slide or two. Reverse or beta turn. So these are essentially turns in your primary amino acid sequence. Sometimes they're supported by things like metal ions. Sometimes they're just supported by interactions with the solvent around them. And then lastly, one we don't talk too much on, but some more information is contained in your book, a secondary structure called the random coil, which is essentially a coil that helps stabilize the tertiary structure of proteins, right? Again, they are critical to completing the structure of a protein, but if you want to learn more about those, either come ask or you can refer to the textbook. Oop. So some more comments about the alpha helix, right? So as the name implies, alpha helixes are secondary structures of tightly coiled helical shapes or strings of amino acids that, when aligned, can form to make the shape of a rod. And one of the primary reasons these fold and are stable is because as our string of amino acids coil around each other, like in our example over here, the carboxyl groups and amine groups between amino acids in this section of the coil and this section of the coil actually form hydrogen bonds to stabilize this overall shape. Right, so hydrogen bonding can play a large role in stabilizing alpha helices, right? And I was meant to, in lecture, I would have asked a question. So hydrogen bonds between the carboxyl and the amine groups of amino acids help stabilize these. So maybe actually you could take a second here now. So we're talking about how amino acids from one portion of a helices interact with other portions of amino acids from another section of the helices. Maybe take a second to pause again and think about what kind of amino acids might favorably fit into this sequence of amino acids, and then maybe which amino acids don't favorably fit. Right? So on that note, amino acids like proline and glycine typically don't find themselves in an alpha helices. If you remember back to the first time we discussed proline, proline is a fairly rigid amino acid because of the cyclic structure that forms from its R group, right? So it's the nature of an alpha helices to curl and curve, and sticking in a very rigid amino acid like proline would likely stop the ability of an alpha helices to coil and turn. Glycine is also slightly less favorable because of that R group that's only an H. Right? And then, so on that note of how amino acids are playing larger roles, so the tendency to incorporate amino acids in alpha helices varies according to the R group and order of residues, right? So in addition to proline and glycine not being favorably found in alpha helices, or more accurately, favorably attributing to stability of alpha helices, amino acids with big, bulky side chains also typically aren't very favorable, right? And so you can't see that too well in this cartoon over here, but in this GIF of a rotating amino acid, we have our hydrogen bonds here, again, between that carbonyl and our amino groups. And then if you look closely, you can see that this stick here, which is meant to represent the R groups found on amino acids, the R groups in an alpha helices typically point outward, right? So speaking from a stable and low energy perspective, you wouldn't want R groups to be sticking into the inner core of our alpha helices, because that would destabilize and disrupt the shape. So typically on the outside of these alpha helices, there are R groups, and depending on what environment these alpha helices are found in, those can be either hydrophilic or hydrophobic, right? Remember that proteins have evolved over time to essentially have hydrophobic R groups interact with other nonpolar regions of a cell, so like the interior sections of a cellular membrane, or if the job of this alpha helices was to reside in the cytoplasm where there's a lot of water, maybe these are chock full of hydrophilic R groups. So if you have a slightly more linear mind, right, maybe if three-dimensional structures don't help too much, so this is meant to be a linear representation of those hydrogen bonds. So if you recall from the previous slide, typically these helices have 3.6 amino acids per full rotation, right? So that would mean if we stack these all up in that three-dimensional structure, the carbonyl from this amino acid forms a hydrogen bond from the amine of this amino acid roughly 4 amino acids away, right? And so if you scroll backwards, you'll again see that these amino acids are forming hydrogen bonds up and down the axes of our helices. So this guy would form a hydrogen bond with this guy because he's stacked on top. And here's just a slide to demonstrate, the favorable or unfavorable energetics of including amino acids, specific amino acids, into these alpha helices, right? So the title of this table is the propensity of amino acids to take up an alpha helical conformation, and the data here says that it's normalized to alanine, right? So delta delta G naught is the difference in free energy change relative to that for alanine required for the amino acid residue to take up the alpha helical formation. So if you look up an alanine, the delta delta G naught is 0. So this would mean that low numbers here indicate amino acids that are favorably found in in alpha helices. So if we look closely, alanine 0, arginine 0.3, and then if we go to and point out those two amino acids I mentioned earlier, right, glycine and proline, have delta delta G naught values of 4 or greater than 4. Again, indicating that it's energetically unfavorable to include glycine and proline in alpha helices. So another secondary structure, again, that I mentioned earlier, would be beta pleated sheets, right, or beta sheets. So these are large segments of amino acids that extend in a zigzag structure, hence the pleated sheet name, and essentially align in these normally sheet-like flat structures, right? There's two types of beta sheets, right? Parallel and anti-parallel. Those will come up on the next slide. And so maybe take a second here to think about what helps aid keep beta sheets stable So components of amino acids that help stabilize these guys and make them energetically favorable. And then also what some common amino acids in a flat string-like shape like this might be. So again, take a pause here. Okay, hopefully you've thought about that a little bit. So previously we discussed how glycine and proline were typically unfavorable to be included in alpha helices because amino acids like proline add rigidity to helices, where a helices is a curled coiling structure. But in the case of beta sheets, right, they're these long, flat, string-like things. So adding a rigid amino acid like proline maybe isn't so energetically unfavorable. And if you look closely up here, and or you thought more about it a little bit, a stabilizing interaction that helps keep our beta sheets together is, again, hydrogen bonds, right? Hydrogen bonds between those amino and carbonyl groups. They help keep these guys together and compact and stuck close together. So noted here, right, we have an example of an anti-parallel beta sheet. So anti-parallel refers to the direction of C-terminus to N-terminus flow of amino acids, right? So anti-parallel is when sheets that are next to each other essentially go C-terminus to N-terminus and then loop back and go C-terminus to N-terminus. And again, here in this image and this image, you can see how, again, those carbonyl groups and amino groups are interacting to form hydrogen bonds, which adds stability to how these sheets lay out. And then a note about R group organization. So previously when we mentioned where R groups find themselves in alpha helices, similarly to an alpha helices, our R groups aren't going to find themselves in between the strings of amino acids, because that would introduce steric hindrance and a number of other things that would decrease the stability of these secondary structures. So typically, R groups in beta sheets extend upward and downward, right? Almost in a trans organization, if you were to think about these as long, long planar views of covalent bonds, right? So R groups typically go above and below these sheets. So if an anti-parallel beta sheet runs C-terminus to N-terminus, folding back on C-terminus to N-terminus, what do you think a parallel beta sheet's going to look like? A parallel beta sheet is going to run C-terminus to N-terminus and then match up again with C-terminus to N-terminus, right? So recall our C-terminus and N-terminus nomenclature from previously in the semester, and it's just a different formation from the anti-parallel. We won't get into the specifics of which is necessarily more favored or more functional. So just some notes on characteristics or structures that don't really fit into the secondary structure nomenclature. So we refer to these as super secondary structures, and they typically form larger functional motifs or larger functional domains, right? So some examples here. Some examples are shown here. So we have, an alpha heli here, a beta meander, a beta barrel, right? So a beta barrel forms when multiple parallel beta sheets align and form a pore, right, or an open hole in the center here. And ultimately, the function of these domains or motifs comes down to what the specific protein is responsible for. So we've discussed sulfhydryl bonds or disulfide bridges a number of times throughout the semester, but I'm just going to include another little note and slide here, right? So recall that sulfhydryl groups, these sulfhydryl groups or thiol groups of cysteine residues can be, depending on the environment, highly reactive and actually form bridges between different areas of large peptides or large polypeptides, right? So over here we have our unbonded cysteine residues, and again, depending on the environment that these guys are found in, they can either remain non-bound to each other or bound to construct those disulfide bridges. So if the environment they are found in is a highly oxidative environment, right, these two functional groups can be oxidized, which will result in the formation of that disulfide bridge, which can add to I'm going to stick to the beyond secondary structures here, because this may, depending on who you ask, this might be considered tertiary structure, or if this is the formation of this disulfide bond is a component of tertiary structure, right? If it's bonding two polypeptides together, it's a little gray. Again, I'm going to keep emphasizing that biochemistry and the topics we talk about aren't completely black and white. And then conversely, right, if a disulfide bridge is formed and this section of a protein or polypeptide polypeptide finds its way into a drastically reducing environment, or a reducing agent is added, you can actually break that disulfide bond. And we're going to actually exploit this in one of our activities in lab. We're going to add a reducing agent called beta-mercaptoethanol to help break disulfide bonds, which is going to help us linearize or denature our proteins when we get to our protein analysis section. And our last example for this slide deck for beyond secondary structures is a structure referred to as a leucine zipper, right? So a leucine zipper forms when two alpha helices are found within close proximity to each other, and amino acids like leucine or valine, which are typically and primarily nonpolar, can actually come together and through the exclusion of water form water exclusion properties that allow these nonpolar groups to clump up and gain some form of intermolecular force, right? Most likely van der Waals forces will help keep these two guys together. And I like to use this example as well because it's fairly common and easy to use things like electrostatic charges to discuss how proteins come together and are held together, such as how our alpha helices and beta sheets gain stability. But other structures can form from the exclusion of water, again, and through interactions of nonpolar side chains, right? So I spoiled that there. The type of bonding holding these guys together would be van der Waals forces typically, right? Because there's no formal charges or dipole-dipole moments aiding in the congregation of these side chain R groups on these amino acids. And then a note about the function of leucine zippers. So sometimes leucine zippers are structures found in proteins that help repress the transcription of DNA, right? So sometimes these functional groups are found in proteins or proteins that need to be activated or removed from DNA to help allow DNA to transcribe. Oh, yep, I just gave that away as well. So yeah, sometimes this structure is a component of repression proteins. So again, as always, please feel free to email me with questions if you have any, or come to office hours, right? I'm always available. No matter what time of the day, feel free to shoot me an email.